Very warm welcome to everyone, and it's my pleasure to introduce today's Precision Nanosystems TeaTime webinar for September. I'm very pleased to introduce our speaker today, who is Dr. Federica Sebastiani from Malmö University in Sweden. And Federica will be talking today on can mRNA-Lipid nanoparticle surface composition regulate Apolipoprotein binding from serum. So just before we get started, just a couple of introductory pieces of information and announcements. So firstly, I'd just like to introduce your Precision Nanosystems Europe team. So I'm Richard, Field Application Scientist for the North of Europe, based in Oxford in the UK. And I work with my colleagues, Sarah, who is also a Field Application Scientist based in Paris, and Martin Rabel, who is based in Leipzig in Germany. We also work alongside our sales team, so my colleagues AJ and Suha are both based in London, and Jürgen is based in Berlin, also in Germany. So with that, it's my great pleasure to hand over to our speaker today, Federica Sebastiani, and looking forward to her presentation. Thank you.
Good afternoon, everybody. I'm very happy to have this opportunity to talk about my work. And so as Richard already said, I'm going to talk about mRNA-Lipid nanoparticles and what happens when they bind to apolipoprotein. I would like to say that this work was possible thanks to the great collaboration between academia and industry, in particular with Marianna Janez Arteta in AstraZeneca. And I think it's a great example of how academia and industry can collaborate to produce good science. So let's dive in. Why mRNA therapeutics? I guess this audience knows very well why, but I would like to stress that, well, potentially this is a therapeutic that can treat any disease. And we have seen in the recent time that this was successful as a vaccine for COVID-19, for example. And why it's so powerful? Well, mRNA therapeutics takes the advantage that the messenger RNA can teach our cell to produce any protein that we need, which probably is not possible to synthesize outside the cells. So this great promise brings a lot of challenge as well. You know that RNA is a negatively charged molecule, a long one, that is going to be almost impossible to cross the cell membrane and even arrive to the cell membrane without being degraded by enzymes. So we do need a biocompatible vehicle to breach it in, allow endosomal escape, and deliver the mRNA to the ribosome and produce the desired protein. Now, we selected as vehicle LNPs, and LNPs stands for lipid nanoparticle, which is made of different lipids and cholesterol. In particular, in this work, we selected MC3, a cationic ionizable lipid that is the key component that allows to complex with the messenger RNA and protect it from the environment. And then we have other three components that help the particle to have the right size and be stable and so on. So our collaborator in AstraZeneca, in particular Marianna Janez, did a great job on looking at the structure of these lipid nanoparticles using scattering. And what it was great about this work is that they linked the structure to the function of the particle. So what they found out is that the lipid, the helper lipid, the DSPC, is mostly at the shell of this particle. So this particle has some kind of core shell structure, and the DSPC is mostly at the shell. It's not homogeneously distributed in the particle. And what they found out is that if you if you keep the area per molecule, per DSPC molecule, constant, increasing the size of the lipid nanoparticle, you get an improved expression of protein, given the same dose of mRNA. While if you don't pay attention to keeping this surface constant, so you just increase the size, this increase in protein expression doesn't happen. So this told us that the surface structure is actually key to function. So the physicochemical characterization of this particle, it's really important to understand and make a step forward to tune the efficacy of these particles. So starting from this work, we decided to look into more details, where the other components were going and what kind of effect could have on the function. And to do that, we used the small angle neutral scattering and contrast variation. So for those of you that are not familiar with that, I will give a brief introduction. This is a technique that allows to study a sample in solution. So you don't need to dry or freeze the sample. It's just in solution. And you, from the scattered intensity, so you have an incident beam shining through the sample, and then you collect the scatter intensity in a 2D detector. You can then do a radial average and you get this kind of graph, so an intensity versus a Q, a momentum. And from this Sand's curve, you can understand what's the shape, what's the size, and what's the distribution of components of your particle. So it is extremely powerful. On top of that, using neutrons is extremely has a great potential for soft matter, so lipids, protein, and so on, because this cross-section for hydrogen and deuterium is extremely different. So they can be clearly distinguished. And that's very good, because in all these lipids and protein, we have plenty of hydrogen, and we can substitute it with deuterium. And that allows us to make different contrasts and highlight different parts of the particle we want to study. So to put it in practice, the first thing we can substitute is the solvent. So instead of water, regular water, we have water with deuterium, so heavy water, and mixing a different ratio of heavy water in the solvent that we can match, so make invisible the components that we need, depending on the percentage of D2O. And this is the example. So if we have a system that is a core shell, for example, and in water, you have 3 different cross-section, then you can, for example, tune the solvent to have the same scattering length density as the core, and then we'll highlight the shell. You can as well make another sample that has a solvent matching the scattering density of the shell, which will highlight the core contribution. And putting these two information together, you will learn more about the overall system. So, taking advantage of contrast variation, we decided to study the lipid nanoparticle for messenger RNA delivery with this composition, the standard composition that was used by Jan and Sarteta as well. And we instead substituted the components with their deuterated counterparts. And we decided to make 3 different samples, one with matched out cholesterol, so cholesterol that has enough deuterium to be matched in deuterated water, and that allows us to localize cholesterol, both making it invisible or making it stand out from the rest of the particle. Then the other sample was a mixture of matched out cholesterol and deuterated DSPC, and that allowed us to highlight the shell of the particle. And the last one was a composition with deuterated cationic ionizable lipid, and that allowed us to localize this lipid. This is so important to know where it is for the functionality of the particle. So here are the results of the SANS experiment, and you can see the 3 different samples. Each sample was measured in different solvent mixture. So this is the percentage of D2O in the buffer. And what we can say qualitatively is that we have this feature here in the low Q region that suggests a core shell structure. So again, confirming what was found by Janens and co-workers. And then in this middle Q range, high Q range, we have a peak that it's probably due to the internal structure. And the interesting thing about this peak here is that it comes up when the D2O content is higher, when the cationic ionizable lipid is hydrogenated, so is the largest, the contrast between solvent and the cationic ionizable lipid, while is not so visible when we have the deuterated cationic ionizable lipid. So this already tells us that most of the internal structure is made up by the cationic ionizable lipid, so it's a key component in the core. And then, well, fitting this data with a model that combines the core shell sphere and a broad peak to describe this part, we can find out what's the distribution of the different components, since we know exactly what was deuterated in each sample. And putting this information together, we can say that the cholesterol is more concentrated in the shell of the particle, while the cationic ionizable lipid is more concentrated in the core of the particle. And the DSPC, as was already demonstrated, is only at the shell of the particle. So now, knowing what's the distribution of components in the particle, in the LNP, we wanted to make it invisible in a given solvent mixture. Why? Well, we wanted to make it invisible, first of all, to see if there were any domain formation, because we can still, even if the component sum out to have a 0 scattering density, if they are forming domains, they still can have some intensity, some scattering intensity. So that was the first thing, to see if it was possible to completely zero the intensity and make it invisible. And on the other side, we wanted to have this tool as a test tool for looking at the protein binding, and that we will see it later. So we, after some different tests and trial and error, we refined the composition between deuterated and hydrogenated components to make a match out lipid nanoparticle. So these are the molar ratio required of deuterated components in the formulation to make this particle invisible. And here you see we have the intensity of scattering, and we measured different samples in different D2O content to see where the signal was going to 0. And you can clearly see there is a minima here that is about 46% D2O content, where the intensity is 0. And these are the data that we collected. Then you can see the black dot and the gray is the buffer, so no particle, and the black is the particle, the match out particle in the same buffer, and they overlap pretty well. Now you see here there is a protein as well, Apolipoprotein E. And that is invisible as well in this solvent. That's why we decided to go for the 46% deuterated PBS. Why ApoE? Well, the role of ApoE, it seems very important, especially for the cellular uptake in hepatocytes of LNPs. So the mechanism that has been suggested is that ApoE can bind LNPs, and then since it's bound to LNPs and act in the fat metabolism in our body, it can bind and it can let in the LNP in the hepatocytes through LDL receptor binding. And that allows the LNP to enter. Now, in particular, we were interested on this stage. So is the protein just adsorbing to the surface of the particle, or is the protein affecting somehow the structure of the particle? That was our question. And that's why we needed to have the particle that was invisible in a given solvent, and exactly the same solvent as the protein is invisible in. Because if we mix these two components and they just attach to each other without changing anything on the particle, then we should still have a zero intensity. While if something happens to the particle, we should see an increase of intensity because there is a movement of components going on or a change in structure. And that's what we did and what we saw. So an increase in intensity was measured when the ApoE was incubated with a lipid nanoparticle in the match-out composition for 3 hours. Now, this is a tiny increase of signal, so that's not enough to resolve the structural change on the particle due to the protein binding. That's why we looked at the interaction with the protein for the other samples as well. So the sample with deuterated cholesterol, the sample with a mixture of deuterated and DSPC deuterated, DSPC and cholesterol deuterated, sorry, and then the one with the cationic ionizable lipid deuterated. And the black one is the particle without protein, absence of protein in the same solvent contrast, while the blue one is in presence of protein in the, again, 46% D2O content. And you can see that the protein makes a huge increase here in the low Q, especially for these two samples that have a very low intensity, is about, you see the scale, while for this sample, the change is a bit less evident, but there is a change as well. So putting together all the results from the fitting of these curves, we could say that the protein binding to the lipid nanoparticle actually push the cholesterol toward the shell, while the cationic ionizable lipid is moving toward the core of the particle. And that's extremely interesting because all the endosomal escape mechanism is based on the fact that the cationic ionizable lipid in the endosome, where the pH drops, becomes positive, and then it can fuse with the endosomal membrane that is negatively charged. But if this effect is not reversible, and when the particle is in the endosome, doesn't have enough cationic ionizable lipid charged positively, then we don't have a good endosomal escape, the fusion with endosomal membrane. And that may be the reason why the efficacy is so low, and the LNP doesn't really release the mRNA in the cytoplasm. So these results just told us about the overall structure and the distribution of the component, but we wanted to look a bit more on the internal structure as well of the particle. And that's what we did with small angle X-ray scattering. And here we have the lipid nanoparticle measured at different time points interacting with ApoE. And this peak is the peak that arise from the internal structure of the lipid nanoparticle, which was proposed to be a kind of disordered hexagonal phase, so channel, water channel where the mRNA is located, surrounded by the cationic ionizable lipid. And here what we can see, the blue up here, is the lipid nanoparticle just as soon as it was mixed with the protein, and then it was followed at 3 hours and then 15 hours from mixing. And you can see that already at 3 hours, the red curve, the intensity drops. So the scattering intensity drops because the structure is probably less ordered than the beginning. So there is not only so this redistribution of components does affect the internal structure as well. And there is a tiny shift as well on the position of the peak toward higher Q, which translates to lower despacing. So the channel distance, so the interchannel distance is shrinking, meaning that maybe you are losing mRNA, which aligns with what this encapsulation efficiency tells us. Here we have encapsulation efficiency and hydrodynamic radius measured at day 0 for different weight ratio of ApoE to mRNA. If we look at the ratio 1, which is the ratio we used for all the Sachs and Sons experiment, for example, at day 0, we already have a drop compared to very low amount of ApoE. So the ApoE has already an effect on the encapsulation efficiency for mRNA. And then after 1 day, there is almost a 20% drop in efficiency. So this redistribution of components, loss of internal structure, actually makes the mRNA leak out from the system, and even more after 3 days. And then, of course, is the effect is kind of increasing with increasing ApoE weight ratio to mRNA, while the overall size doesn't really change so much, which was found as well from the SANS data. So that tells us that the ApoE actually has an effect not only on the structure, but on the encapsulation efficiency as well. And that's something it's important to bear in mind. So I would like to conclude, summarizing. We were able to resolve the component distribution in the LNPs thanks to the use of scattering, neutron scattering and contrast matching. And we were able to look at the effect that ApoE has on the LNP structure itself. And in the future, well, we are working on looking at what's the effect of pH on the structure of lipid nanoparticles. So looking a bit more at this stage of the mechanism of uptake, and that both in present and absence of ApoE. And we are looking at different formulation as well to see what's the effect of the formulation on the distribution of components. And of course, we are interested to look as well on the what's the effect of ApoE on the LNP with different formulation. So I would like to thank the people that contributed to this work, and in particular Marianna Yanez Arteta, that was my collaborator in AstraZeneca, and the instrument scientist that made sure that everything runs smoothly during experiments, and all the people that work at the deterioration of components, which allowed the isotopic the isotopic labeling of the lipids. And last but not least, the funding agency that funded the project. And I would like to thank you for your attention, and I'm happy to answer questions.
Yeah, thanks a lot for all these insight, for your talk. It was really great. Thanks for your time, and thanks for everyone for coming today, and see you at the next TeaTime in 4 weeks' time. Goodbye, everyone.
Thank you.